The second objective here, very briefly, is to talk about a few toxins and how they affect the neuromuscular junction. So the toxins we're going to talk about, the first one is called latroxin. And latroxin is a toxin that is found in the venom of a black widow spider. So if you are ever bitten, well, I hope none of you are ever bitten, but if someone is bitten by a black widow spider and they aren't exposed to help soon enough, these patients will exhibit certain symptoms because of the venom that is in that, the toxin that is in that venom. So the action of this toxin is basically to increase the amount of acetylcholine that is released at the neuromuscular junction. So if we sort of dump extra acetylcholine at the neuromuscular junction, what do you think the clinical symptom would be in this patient? How do you think this would present, given what we talked about the neuromuscular junction?
Paralysis, almost like stood up.
Would it be paralysis? Think about it. So we have acetylcholine. Muscle spasm. Right. So it'll be instead of lack of muscle contraction, it'll be increased muscle contraction, which is called spasticity or muscle spasms and rigidity. So spasticity, muscle spasms, and rigidity is how these patients typically present, because if we have excess acetylcholine, we're going to continually stimulate that motor end plate. We're going to continually contract the muscle. We're going to continually have end plate potentials. Okay, that's latroxin. Let's look at chrotoxin. Chrotoxin is in the venom of a rattlesnake. And this toxin basically blocks the effect of acetylcholine at the junction. So instead of what we saw with latroxin, here we have no acetylcholine at the neuromuscular junction. What do you think that would present with? Muscle weakness, so paralysis or muscle weakness, which is the opposite symptom of latroxin venom. Okay, and then finally there's curare, and curare is found in a plant, and it's typically used by Native Americans or Native Indians, and they put it at the end of their darts when they're hunting to paralyze animals or other threats. And the action of curare is to block this nicotinic receptor. So if we block this nicotinic receptor, we're going to have the same effect as if we had no acetylcholine at the junction, right? Because the acetylcholine, even if it's there, if it cannot bind to this receptor, it cannot stimulate the motor end plate. It cannot generate an end plate potential. And so we're going to have zero muscle contraction, which presents with weakness. And eventually these can become fatal. So chrotoxin, curare, and latroxin all become fatal because of their influence on the respiratory muscles, right? If you have spasm of the respiratory muscles, or if you have weakness of the respiratory muscles, you will pretty much impede your respiratory function, and then that's how that becomes fatal. Okay. Another thing I want to point out is myasthenia gravis is an autoimmune disease. I think we talked about it a little bit earlier, but the way this disease happens is basically the same phenomenon as curare. So myasthenia gravis, the body makes antibodies against these receptors. So it pretty much destroys these receptors. And so we have that same symptom of muscle weakness and paralysis as we do with the curare toxin. So the treatment for myasthenia gravis is to block the acetylcholinesterase receptors. And how do you think that would affect these patients? So let's say you had muscle weakness because of the curare toxin, or you had muscle weakness because of myasthenia gravis, where your body destroyed those receptors. How would blocking acetylcholinesterase fix that or give those patients some relief? How do you think that plays a role in this phenomenon? So what is the function of acetylcholinesterase? So it breaks down acetylcholine, right? Excess acetylcholine, and when does it really act? What's the function of it? So when we're ready to cease muscle contraction, right? When that end plate potential is over, acetylcholinesterase clears away the excess acetylcholine at the junction. Now, if you have a patient who has fewer receptors, fewer nicotinic receptors, or damaged nicotinic receptors, you're basically not going to get muscle contraction as effectively. So would you want to clear away excess acetylcholine from the junction? You would not, right? You'd want that acetylcholine to stay around as long as it could to interact with whatever receptors are working or to interact in any way that it could to help to preserve whatever muscle function that you could preserve. Okay, so the clearing away of the neuromuscular junction that acetylcholinesterase helps to bring about is not necessary in patients who have damage to these receptors. In fact, it makes the disease worse. So when we block these receptors, these patients improve drastically. And it's like a night and day from the time they receive this therapy, where they can suddenly get muscle function simply because we're blocking these receptors. These receptors are working against us if we don't have adequate function of, these enzymes are working against us if we don't have adequate function of the receptor.